Once a bioinformatics pipeline has identified a patient's genetic variants, the crucial questions we need to ask are, what do these variants mean, and how can we translate this data into a targeted clinical decision? The answer lies in the vast world of omics databases, which serve as a bridge between simple biological information and clinical insight. The final output of the bioinformatics work is a list of millions of genetic variants, and this is where the annotation and prioritization phase comes into play. The analysis of a variant, whether it's a mutation or a polymorphism, must determine three key elements: its pathogenicity, that is, whether it causes or contributes to the disease, its frequency in the population, and its pharmacological action, meaning whether there is a drug that can inhibit or leverage its effect. Only the variants that pass this filter, often compared with reference clinical databases like ClinVar or Cosmic for oncology, are promoted to the status of therapeutic biomarker. This biomarker is the molecular key that unlocks personalized treatment. Let's see how these and other biomarkers are specifically used to guide the selection of targeted drugs in precision oncology. There are several categories of databases that host aggregated data of various kinds. Let's start with population databases, like the 1000 Genomes Project and the NOMAD Genome Aggregation Database, which have revolutionized diagnostics. These databases aggregate sequencing data from hundreds of thousands of individuals from different populations and serve as a critical causal filter. Without them, every rare and non-patient-specific variant would have to be studied from scratch, making the diagnosis of rare diseases an unsustainable, lengthy, and expensive process. In this context, the first step is to analyze the allele frequency in NOMAD, which allows us to rule out common variants, and they are probably benign, focusing attention on those rare ones that are more likely to be pathogenic. For example, if a variant has an allele frequency of 5% in GNOME-AID, it's highly unlikely to be the cause of an extremely rare disease. For clinical practice, there are more specific resources that directly associate variants with diseases. It's important to keep in mind that these resources are constantly evolving. Let's look at a few non-exhaustive examples. The first database I want to mention is ClinVar. This is a curated database that links genetic variants to clinical conditions and published studies. It provides an assessment of pathogenicity, for example, classifying them as pathogenic, benign, or of uncertain significance. Two other fundamental databases are Cosmic and the Human Somatic Mutation Database, HSMD, which are used in oncology to link cancer somatic mutations to clinical and scientific information, and information related to drugs. The Human Somatic Mutation Database can, for example, indicate that a specific mutation in the EGFR gene in lung cancer is a potential therapeutic target for a specific drug. These tools, therefore, translate mutations into potential therapeutic targets for the development of new drugs and clinical decisions. In Italy, as in the rest of the world, an innovative clinical approach is spreading based on molecular data that allows, among other things, treating a tumor with a drug originally developed to cure a completely different type of cancer. This happens because the biology of cancer is increasingly defined by somatic genetic alterations, mutations, in other words, rather than just by the organ where it appears, like the lung or the colon. If a specific genetic mutation that responds to a certain therapy is identified in a new type of tumor, it is possible to obtain authorization to use that drug for treatment. This process is called drug repurposing or drug repositioning. It means applying a drug that has already been approved for another disease is advantageous because repurposed drugs have already passed safety tests, which speeds up patient access to targeted therapies. When the variants are not present in these more clinically oriented databases, or when we are analyzing a model organism, it is useful to broaden the range of useful databases. The NSPI ecosystem and similar interconnected resources like Ensemble and UCSC are the reference databases, and they are entirely public. These databases are not focused on pathology, but they offer a vast catalog of gene sequences and annotations, including raw data from experiments. We can think of them as an interconnected library. GenBank is the primary database that holds the collection of all publicly deposited DNA sequences. SRA is the main archive for raw sequencing data and is connected to YANA, its European counterpart. Closely linked to raw data, we find Gene Expression Omnibus. In this repository, you'll find the results of analyses of SRA raw data, as well as the metadata that characterize the study. This resource becomes crucial for preliminary exploration, searching for validation datasets, or for meta-analysis studies useful for identifying biomarkers. If we're looking for genomes and exomes, since these data are protected by controlled access policies, we can turn to DBGP, the database of genotypes and phenotypes. We also have DBAR and DBSNP, which catalog structural variants and known polymorphisms, respectively. All this knowledge and many other annotations are integrated and made available through the use of visualization tools known as genome browsers, including UCSC and Ensemble. Genome browsers allow you to explore biological data in a spatial and functional context, integrating complex data to draw definitive conclusions, for example, by visualizing where the ClinVar variant is located within the gene under examination. By leveraging these incredibly vast and comprehensive resources, specialized computational tools such as VEEP and SNP have been meticulously developed and refined to accurately predict the functional effects of genomic variants. VEEP, or Variant Effect Predictor, developed by Ansible, for example, can predict the effects of variants on transcripts and proteins, but also on regulatory regions and transcription factor binding motifs. These tools make it possible to prioritize which variants to study. It's necessary to broaden our perspective beyond the immediate application in human health, recognizing the cross-cutting impact of genomics. Ambitious projects like the Earth Biogenome Project aim to sequence the genetic makeup of over 1.5 million living species, creating a digital bank of life. This not only provides biological knowledge, but also fuels conservation efforts and the search for new biotechnological applications. Just think about the importance for human health of applications like metagenomics, which is used to study the human microbiome. An in-depth understanding of people's microbiome could open up new avenues for therapeutic approaches. Large-scale sequencing initiatives also exist at regional and national levels. Examples include the LifeMap project, which aims to link pediatric pathological conditions such as obesity, inflammatory diseases, and neoplasms with predispositions to cardiovascular and neoplastic diseases in adults, in order to develop personalized therapy and prevention plans. These actions demonstrate the impact of genomics on public health, providing population-specific data to improve local diagnostic accuracy. We experienced an example of this during the COVID pandemic, when genomics was used to monitor emerging local variants all over the world. In conclusion, a deep understanding of these databases that host population and clinical data, as well as raw data repositories, combined with the ability to use them critically, is at the heart of precision medicine. This process of interpretation is the key to translating population genetics and technical data into concrete clinical recommendations, and to guide the application of genomic data in biology and medicine.